Good morning, everyone. We are from Group 5, and today we will be discussing baseline diagnostics in parasitology, which focuses on the fundamental laboratory techniques that are used to detect and identify parasitic infections. So to begin, parasitic infections cannot be accurately diagnosed based on clinical signs alone. This is because the symptoms are often non-specific and can overlap with many other diseases. Because of this limitation, laboratory confirmation becomes essential. And this is where baseline diagnostics plays an important role. Baseline diagnostics refers to the basic and routine laboratory methods that are actually being utilized as a first step in identifying diseases, especially parasitic infections. So these techniques help us directly detect the parasite, its developmental stages, or its products in different clinical samples. The symptoms of parasitic infection, ranging from chronic gastrointestinal distress to acute episodes, frequently mirror bacterial, viral, or even non-infectious pathologies. And consequently, laboratory confirmation is the requisite standard. The diagnostic process is primarily centered under the direct detection of the parasite's life stages, specifically the ova, the larvae, trophozoites, and cysts. So using high-resolution microscopy and specialized concentration or staining techniques without laboratory evidence, a definitive diagnosis is statistically untenable. The first step in diagnosis is actually selecting the correct specimen. Different parasites can be found in different parts of the body, so proper samples collection is very important. For example, sputum is used to detect lung parasites or larvae that migrate through the respiratory tract. We have the tissue biopsies that help identify parasites that invade body tissues, and even muscle samples that are especially useful for parasites like Trichinella that infect the muscles. For intestinal parasites, the most common specimen is stool, which may contain eggs, cysts, larvae, or even trophozoites. Blood samples are essential for detecting blood parasites such as malaria and microfilariae. Lastly, urine samples can be used to detect parasites like Schistosoma hematobium. One of the simplest and most commonly used methods is the direct microscopic examination. And in here, it actually involves the direct observation of clinical specimens such as stool, blood, or urine under the microscope without extensive processing. It is a quick and immediate method that allows early detection of parasitic forms like trophozoites, cysts, eggs, and even larvae. One of the most basic preparations under direct examination is the saline wet mount. In this method, a small amount of stool is mixed with a saline solution on a glass slide and covered with a cover slip. It is then examined under the microscope. So this technique is especially useful because it actually allows us to observe motile, trophozoites, and larvae in their living state, along with the helminth eggs. Commonly detected organisms in here would actually include our Entamoeba histolytica, their trophozoites, the Giardia lambleya, their trophozoites as well, and the eggs of Ascaris lumbricoides. Another important wet mount is the iodine wet mount, primarily used to enhance and visualize the internal structures of protozoan cysts, such as the nuclei and organelles. Instead of saline solution, in here we utilize a Lugol's iodine. The stain does not actually preserve parasite movement, but rather it enhances internal structures such as nuclei and organelles, making protozoan cysts easier to identify. Notice in here, in the photo, as you can see in here, there is a distinct yellow-brown coloration in these 4 microphotos. While the iodine kills the parasite, it acts as a stain that highlights internal structures like nuclei and organelles. So this is what allows us to accurately identify the specific species of a protozoan cyst. Some examples of the organism that can be detected, again, is Entamoeba histolytica, the Giardia lamblia. Moving on to concentration techniques, we have the sedimentation method, specifically the formalin ether technique. This method works on the principle that parasite eggs and cysts are heavier than the surrounding fluid, so they naturally settle at the bottom of the tube after centrifugation. This allows for better recovery and concentration of parasites from stool samples, increasing the chance of detection of parasites such as Ascaris lumbricoides eggs and Trichoris trichoria eggs. So if you look at the 4 vials in the image, we can actually see in here that there is a separation of the layers. So pointing to the last vial, we can see that there is a dark pellet settling at the bottom. The tiny sediment contains the concentrated eggs and cysts from the entire sample, making it much easier to detect light infections. In contrast to sedimentation, the flotation technique uses a dense solution such as the zinc sulfate or saturated salt. Here, lighter parasitic eggs and cysts float to the surface while heavier debris sinks. The material collected from the top layer is then examined microscopically, so this method provides a cleaner background, making it easier to identify parasites clearly by initially mixing stool samples with flotation solution centrifuge to separate the debris. Parasites float then to the surface, and we will collect this. For more detailed examination, we used permanent staining techniques. It is used to detect and confirm the presence of intestinal protozoa by enhancing their structures and morphology. So this actually involves preparing a thin smear of stool, fixing it, and then applying a series of stains. The stained slide is then examined under oil immersion. This technique is very useful for confirming protozoan infections because it highlights internal structures that are not visible in wet mounts, like Entamoeba histolytica and Giardia lamblia. Most commonly used permanent stain is a trichrome stain. This is useful for detection of intestinal protozoa and stool specimens by enhancing their cellular structures. This stain provides excellent contrast, usually coloring parasites in shades of purple or red against a green or blue background. This makes it easier to differentiate parasites from debris such as yeast or plant material. The procedure in here would begin by preparing, of course, a smear, applying a trichome strain, and then rinse and examine under a microscope. So the striking contrast, as you can see in the photos, there is a purple or red stained parasite against a green or blue background. This color differentiation is vital because it allows us to instantly distinguish a real parasite from artifacts like yeast or vegetable fiber. So this helps to detect Entamoeba histolytica, Giardia lamblia, and Entamoeba coli. For blood parasites, we use thin blood smear. This technique is mainly useful for species identification. A single layer of blood is spread in slide, fixed with methanol, stained with Giemsa stain, which helps to clearly show the shape and structure of parasites and blood cells, and they are then examined in microscope. Because in here, red blood cells remain intact, we can observe parasites inside them, such as different species of malaria. This is actually the gold standard for identifying which specific species of malaria is infecting the host. It is very common in Plasmodium species like Plasmodium falciparum, vivax, and malariae. On the other hand, the thick blood smear is used to increase sensitivity. Here, several drops of blood are placed and not fixed, allowing red blood cells to break down. This concentrates the parasites in one area, making it much easier to detect infections even when parasite numbers are low. They are then air dry, stained with a Giemsa stain. However, red blood cells are lyse during staining, making parasites easier to detect and increase their sensitivity. Some of the parasites that we can detect is the Plasmodium falciparum and Trypanosoma species. So in the photos you can see in here, we actually can compare the dark concentrated blood spot to the thin smear because we've cleared away the red cells. We can find parasites much faster. Finally, we have the CATOCADS technique, which is a quantitative method used for stool examination. This technique not only detects helminth eggs, but also measures infection intensity. By calculating the eggs per gram of stool, it also uses a standardized template, a fixed amount of stool, and in here, the stools will be pressed through a template, and they're then cleared and examined microscopically. So this makes it especially useful in epidemiological studies and infection monitoring, which helps detect the presence of Ascaris lumbricoides, Trichoris trichoria, hookworms such as Ancylostoma duodenale, and Nicator americanus. Thank you.
The Harada-Mori technique is a classical parasitological method used to recover and identify nematode larvae from stool samples. Its principle is based on a natural migration of larvae. They move from the stool through moist filter paper into water. The procedure involves 4 steps: smearing of a stool sample on filter paper, 2 placing the paper in a test tube with water, 3 incubating the tube for several days, 4 allowing larvae to migrate downward for microscopic examination. This technique is particularly valuable for detecting hookworm and strongyloides infections. Its strength lies in the simplicity and reliability, making it a cornerstone in stool-based parasitology diagnostics. Culture methods are essential for growing parasites under controlled laboratory conditions. 2 common approaches are protozoan culture and larval culture techniques. Applications include identification of parasite species, observation of parasite development stages, research and drug testing, where cultured parasites serve as models. The advantage of culture methods is that they improve detection of living parasites and allow species differentiation. For example, culturing Entamoeba histologica helps distinguish it from non-pathogenic amoeba, which is critical for accurate diagnosis. Moving into immunology, these tests detect either parasite antigens or host antibodies. Common examples include ELISA rapid diagnostic tests and indirect fluorescent antibody tests. They are applied in diagnosing tissue parasites such as Toxoplasma gondii, screening infections in populations, confirming difficult cases where microscopy fails. Their advantages are speed and sensitivity. For instance, ELISA can detect low levels of antibodies, making it useful for early diagnosis. These tests are indispersible when parasites are hard to visualize directly. Imaging techniques provide a non-invasive way to detect parasites in tissues and organs. Common methods include ultrasound, CT scan, MRI, and X-ray. They are used to detect tissue parasites, assess organ involvement, identify cysts and lesions. For example, ultrasound can reveal hydatid cysts in the liver caused by Echinococcus granulosus. CT and MRI scans are particularly useful for neurocysticercosis, where cysts form in the brain. These techniques not only confirm infection but also guide treatment decisions by showing extent of damage. Rapid diagnostic tests, or RDTs, are field-friendly tools that provide quick detection of parasite antigens. They use immunochromatographic test strips. The procedure is straightforward. A blood or sample is applied to the kit. Reagents migrate along the strip. A colored line indicates a positive result. Advantages include speed, ease of use, and minimal equipment requirements. Results are available within minutes, making RDTs especially valuable in remote areas. For example, malaria RDTs are widely used in endemic regions where laboratory facilities are limited. PCR is a molecular diagnostic method that detects parasite DNA or genetic material. Its principle is amplification of small amounts of DNA, making even minute traces analyzable. Applications include species confirmation, detection of low-level infections, and epidemiologic studies where genetic markers are tracked. The advances are high sensitivity and specificity, ensuring accurate identification of parasites. However, limitations include expense, the need for trained personnel, and specialized equipment, which restricts its use mainly to advanced laboratories. Beyond blood and stool, other body fluids can reveal parasitic infections. Sputum examination, used to detect Paragonimus westermani eggs, which appear as brownish iron fillings streaked with blood. Urine examination, concentrated sediment may show schistosoma hematobium eggs with a terminal spine or Trichomonas vaginalis. Chylous urine, examined for microfilariae in lymphatic filariasis. These examinations highlight how parasites inhabit diverse niches in the body, requiring tailored diagnostic approaches. Biopsy methods provide definitive diagnosis by directly sampling tissues. Muscle biopsy detects Trichinella spiralis larvae in taeniosolium cysts. Skin snip. A small cone of skin is sliced to detect subcutaneous parasites like Onchocerca volvulus and Leishmania. Tissue or lymph node aspirate. Demonstrate Leishman-Donovan bodies in visceral leishmaniasis. Though invasive, these techniques are invaluable when parasites reside deep in tissues and cannot be detected by less direct methods. Enterotest or duodenal capsule technique. This non-invasive method involves swallowing a gelatin capsule containing nylon yarn. Once dissolved, the yarn reaches the duodenum and collects bile-stained mucus. After revival, the sample is examined microscopically. It is highly effective for detecting Giardia lamblia trophozoites, Strongyloides stercoralis larvae, and eggs of liver flukes. Enterotest is particularly useful for diagnosing parasites in the upper intestine without requiring invasive procedures. Intradermal skin test and in vivo methods. These methods rely on host immune reactions or vector biology. Canzoni test involves intradermal injection of hydatid fluid to diagnose hydatid disease caused by Echinococcus granulosus. Leishmanin or Montenegro test measures delayed hypersensitivity positive in cutaneous leishmaniasis. Cynodiagnosis, an uninfected vector such as red feet bug feeds on a patient and its feces are later examined for Trypanosoma cruzi. These approaches are unique because they combine immunologic principles with biological interactions, offering diagnostic options when conventional methods are insufficient.
Hello everyone. Right now we're going to talk about a very important topic in diagnostic parasitology, the quality assurance or QA. A robust QA system ensures that every step from sample collection to result reporting is accurate, reliable, and timely. Let's break down why this matters so much in parasitology and how laboratories in the Philippines maintain high standards. To begin with, quality assurance is a systematic program designed to guarantee reliability across all testing phases. Now, why is QA especially significant in parasitology? First, many parasites are structurally fragile. For example, trophozoites can under lysis within just 30 to 60 minutes after collection. Second, diagnosis largely depends on microscopy, which carries a high risk of operator error. As a result, misdiagnosis can lead to serious consequences, including patient death. Therefore, implementing strong QA measures is not just a best practice, it's a life-saving necessity. Quality assurance typically involves three main stages: the pre-analytic, analytic, and the post-analytic. Pre-analytic phase covers specimen collection, labeling, and transport. The analytic phase includes actual microscopic examination and identification. Finally, the post-analytic phase ensures accurate result reporting and interpretation. Each stage must be carefully monitored to prevent errors that could compromise patient care. Moving on, let's discuss the National External Quality Assessment Scheme, or NICAS. This is the Philippine National EQA program managed by the Research Institute of Tropical Medicine, or RITM. NICAS evaluates laboratory performance by assessing staff proficiency and testing integrity from sample receipt to result release. Importantly, participation in NICAS is mandatory under local regulatory policies. Without it, a laboratory cannot obtain or renew its license to operate. Moreover, the RITM has been officially designated by the Department of Health as the National Reference Laboratory for Malaria and Other Parasites since 2000. RITM administers NICAS by sending specimen panels to laboratories and evaluating their performance using a system called OASYS. Since 2018, NICAS participation has been a requirement for DOH laboratory licensure. A 2019 study in the Philippine Journal of Pathology showed that NICAS effectively identifies diagnostic gaps, such as difficulties detecting Schistosoma japonicum and accurately estimating malaria parasite density. Now, let's focus on a specific internal QA technique, the blinded cross-checking method. The term blinded means the second examiner does not know the first examiner's result. Cross-checking means comparing results from different examiners or laboratories. In practice, a senior microscopist rereads a defined percentage of positive slides and all negative slides without knowing the original findings. This prevents false negative and false positives, both of which could compromise patients' care. So we have here a diagram of the blinded cross-checking method and a simple comparison table. As you can see in the table, if both examiners report positive, the result is concordant. If the first reports positive and the second is negative, that's discrepant. Any discrepancies are thoroughly investigated and corrective actions are implemented when necessary. Finally, the blinded cross-checking method is commonly used in tuberculosis smear microscopy, malaria diagnosis, HIV and serological testing, clinical microbiology laboratories, public health surveillance programs. In summary, this method is an essential component of laboratory quality assurance systems. It helps maintain high diagnostic standards, ensures dependable results, and supports effective disease management and public health monitoring. Thank you, and that's all for my part. But remember, in diagnostic parasitology, quality assurance is not just about protocols. It's about protecting patients and improving health outcomes. Stay precise and stay vigilant.
Good day everyone, my name is SONICO, and for this part of the presentation I will be talking about rapid diagnostic tests or RDTs. Now, what are RDTs? RDTs are point-of-care diagnostic tools designed to quickly detect infections without the need for complex laboratory equipment. Now, they are commonly based on immunochromatographic or lateral flow principles, where an antigen-antibody complex reaction produces a visible colored line on a test strip. Now, in simple terms, when a sample such as blood or urine is applied to the test, it flows along a membrane. Now, if the target antigen or antibody is present, it binds to labeled antibodies and forms a visible line indicating a positive result. Now, one of the biggest advantages of RDTs is their speed and accessibility. Results can be obtained within 15 to 30 minutes, making them highly useful in remote or resource-limited settings where laboratory facilities are not available. However, despite their convenience, RDTs also have limitations. Their accuracy may be lower compared to more advanced methods like PCR, and some tests cannot distinguish between current and past infections, which may affect clinical interpretation. There are many kinds of RDTs, but for today we will only be focusing on two types, which are the malaria and schistosoma RDT, which are the two top parasitic diseases in our country. Now, malaria RDTs are used to detect specific antigens produced by Plasmodium parasites in the blood. Now, these tests are especially useful in areas where microscopy is unavailable or when immediate diagnosis is needed. Now, there are two main antigen targets commonly used in malaria RDTs. The first one is the PFHRP2 or the histidine-rich protein 2. This antigen is produced especially by Plasmodium falciparum, which is the most dangerous species causing malaria. Because of its high sensitivity, PHFRP2 is very effective for detecting P. falciparum infections. However, one limitation is that this antigen can remain in the bloodstream even after successful treatment, causing false positive results. The second one is the PLDH or Plasmodium lactate dehydrogenase. Now, this enzyme is produced by all species of Plasmodium and reflects active infections. Since it is only present when parasites are alive, once treatment is successful and parasites are cleared, PLDH levels drop quickly, making it useful for monitoring treatment response. Now, some modern RDTs combine both types now to improve detection accuracy and species differentiation. Now, it is important to note that some P. falciparum strains have PFHRP2 gene deletions, which may lead to false negative results. Now, this highlights the importance of combining diagnostic approaches when possible. Lastly, we have the schistosoma rapid diagnostic test, specifically the circulating cathodic antigen or CCA test. Now, this test detects CCA, which is a glycoprotein antigen released by live adult schistosoma worms. Now, the antigen circulates in the bloodstream and is eventually excreted in urine, which is why this test uses a urine sample instead of stool. Now, the CCA test is performed using a dipstick method, similarly with the other lateral flow assays, and results can be obtained quickly and easily. One of the main advantages of this method is that it is non-invasive, since it only requires urine collection, and it is also faster and more convenient, making it ideal for mass screening programs in endemic areas. However, the CCA test also has limitations. It may be less reliable for certain species like Schistosoma hematobium, and sometimes results may appear as strays, which can be difficult to interpret. Now, compared to the traditional CATO-CATS technique, which requires stool samples and microscopic examination of parasite eggs, the CCA test is more practical and can detect active infections even in cases with low parasite burden. That's why it is considered as non-invasive and much more reliable than the CATO-CATS technique. In summary, rapid diagnostic tests provide a fast, practical, and accessible approach to diagnosing parasitic infections, although they are best used alongside traditional methods for improved accuracy. Now, that would be all for my part. Thank you.
In this portion, we are stepping away from the traditional view of parasitology, aka the one dominated by glass slides and stain smears, and entering what we call the molecular revolution. While microscopy has been our gold standard for over a century, it has certainly reached its ceiling. Therefore, we are moving toward a future of genomic surveillance where we don't just look for a parasite. We decode its entire identity to understand how it moves through our communities and how to stop it. The first is the loop-mediated isothermal amplification, or LAMP for short. It is an autocyclic strand-displacing DNA synthesis method. And unlike the polymerase chain reaction, or PCR, which requires a thermal cycler to achieve DNA denaturation through heat, the LAMP utilizes the BST DNA polymerase. This enzyme possesses high strand displacement activity, which allows for DNA synthesis at a constant temperature between 60 degrees Celsius and 65 degrees Celsius. The process, as shown in the diagram, requires 4 to 6 specifically designed primers. The inner primers FIP and BIP initiate the process, while the outer primers S3 and B3 facilitate strand displacement. This results in the formation of a dumbbell DNA structure. These structures contain multiple loops that serve as starting points for subsequent rounds of amplification, leading to the exponential accumulation of DNA, often reaching 10 to the power of 9 or billions of copies in less than an hour. The final product consists of stem loop DNAs with several inverted repeats of the target, appearing as a cauliflower structure. The naked eye detection mentioned here is not merely a qualitative observation, but a result of a specific stoichiometric byproducts of DNA synthesis. During the LAMP reaction, the BST polymerase incorporates deoxynucleotide triphosphates or DNTPs into the growing DNA polymer. This chemical reaction results in two significant byproducts. For every nucleotide added, a pyrophosphate ion is released. These ions react with the magnesium ions present in the buffer to form magnesium pyrophosphate, which is an insoluble white precipitate. In high titer samples, this results in visible turbidity, which can be quantified using a turbidimeter or observed visually. The polymerization process also releases hydrogen ions or protons. In a weakly buffered system, the accumulation of these protons leads to a significant drop in the solution's pH, therefore increasing its acidity. To visualize this without the equipment, specific indicators are added to the pre-reaction mix. The phenol red is frequently utilized as it functions as a pH indicator that shifts from red-pink to yellow when the pH drops below 6.8. Alternatively, metal ion indicators like hydroxynaphthol blue or HNB are used. HNB is sensitive to the concentration of the free magnesium ions is depleted by the pyrophosphate reaction. The HNB then shifts the solution color from violet to a distinct sky blue. This chemistry allows for definitive binary, either positive or negative results at the conclusion of the isothermal incubation period. The thermal advantages of LAMP are quantified in the comparison chart. RT-PCR requires high-precision thermal cycling, whereas LAMP maintains a constant temperature, enabling the use of simple heat blocks or insulated water baths. A critical biochemical byproduct of the LAMP reaction is the production of large quantities of magnesium pyrophosphate, which causes visible turbidity. Furthermore, the incorporation of DNTPs into the DNA polymer releases protons, causing a drop in pH. These allow for colorimetric detection using pH indicators such as phenol red or hydroxynaphthol blue. This chemistry eliminates the requirement for gel electrophoresis or specialized optical sensors, facilitating point-of-care or POC diagnostics in resource-limited endemic regions. Deepening our comparison between RT-PCR and RT-LAMP, we must address the thermodynamic efficiency and enzymatic robustness of the two methods. In traditional PCR, a significant portion of the reaction time is dead time, the interval during which the thermal cycler is ramping temperatures between denaturation temperature of 95 degrees Celsius and annealing temperature of approximately 55 to 60 degrees Celsius. The LAMP eliminates this thermal lag. Because the BST polymerase performs strand displacement and synthesis simultaneously at a constant temperature, the reaction enters an autocycling phase. This then leads to a higher rate of DNA accumulation, where the LAMP can achieve billions of copies of the target sequence in 15 to 60 minutes, whereas PCR typically requires 90 to 120 minutes for a similar yield. A critical fact for field parasitology is that BST polymerase is significantly more resistant to inhibitors than the Taq polymerase used in standard PCR. Clinical samples such as stool or crustacean tissue often contain humic acids, polysaccharides, and heme byproducts that neutralize the Taq polymerase. Data also indicates that the LAMP can often be performed in crude lysates, which refers to the samples that have undergone minimal processing, thereby bypassing the need for expensive and time-consuming spin column DNA purification kits. Second is the multiplex PCR, which requires rigorous primer engineering to ensure diagnostic accuracy. When designing a diarrheal panel for parasites like Ascaris, Trichuris, and hookworms, several technical constraints must be met. First, all primer sets, often 3 to 5 pairs, must have nearly identical melting temperatures, typically within 1 to 2 degrees Celsius of each other, so that a single annealing temperature in the thermal cycler works effectively for all targets. Next, primers must be checked via bioinformatics to ensure they do not form primer dimers with each other. In a multiplex tube, the concentration of oligonucleotides is high. If any two primers are complementary, they will then amplify each other rather than the parasite DNA, leading to false negatives. And lastly, in real-time multiplex PCR, each target is targeted or tagged with a different fluorescent reporter. Example, FAM for Ascaris, Hex for Trichuris, and CY5 for hookworms. The qPCR machine must then utilize the optical filters to detect these different wavelengths simultaneously. This allows the clinician to quantify the parasitic load for each species individually from a single 20 microliter reaction. Unlike PCR, which uses specific primers for one organism, metagenomic sequencing involves the fragmentation of all DNA in the sample. Adapters are ligated to these fragments and the entire library is sequenced. To add to that, once the raw sequence data is generated, often gigabases of data, it is processed through the bioinformatics pipelines. The sequences are compared against the NCBI GenBank database using the BLASTN or Basic Local Alignment Search Tool algorithm. This is how the Philippine clade of P. westermani was identified. Targeted PCR might confirm the presence of Paragonimus, but metagenomics identifies the specific single nucleotide polymorphisms or SNPs that distinguish the Mindanao strain from the mainland Asian strains. This level of resolution is required to track transmission routes and identify local intermediate hosts like Isolapotamon sp that were previously unknown to science. We now shift our focus to the case study of Paragonimus westermani in Mindanao. This research represents the first molecular evidence of a new intermediate host for Paragonimus westermani in the Mindanao region. This study was spearheaded by researchers from the Philippine Genome Center of Mindanao and the University of the Philippines, Mindanao. While P. westermani has been documented in the Philippines, most molecular data previously focused on Luzon and Visayas. This case study addresses a critical geographical gap in the national genomic surveillance of foodborne zoonotic trematodes. The visuals on this slide introduce the two primary subjects of the molecular analysis, the freshwater crab, which is the second intermediate host, and the adult trematode, the lung flu. This study further uses a transition from the traditional morphological observation to higher resolution genomic analysis to confirm the presence of the parasite and identify its host species at the genus level. Paragonimus westermani is the primary causative agent of human paragonimiasis, which is a pulmonary dystomiasis. In the Philippines, the Department of Health recognizes 12 endemic provinces. Data cited in the Ricarte study indicates a reported prevalence of 6.6% in Davao Oriental. A core fact highlighted in the paper is the clinical mimicry between paragonimiasis and pulmonary tuberculosis or TB. Both conditions present with chronic cough, chest pain, and hemoptysis, referring to the expectoration of blood. Because the Philippines is a high burden TB country, patients in rural Mindanao are frequently misdiagnosed and placed on anti-TB medication, which is ineffective against trematodes. As shown in the CBC life cycle diagram, P. westermani requires a tri-host system. The first intermediate host is the freshwater snails, specifically of the families belonging to Pachychelidae or Thiaridae. The second intermediate host are the freshwater crustaceans, primarily the crabs or crayfish. And finally, the definitive host, referring to the humans or other mammals like the pigs, cats, and dogs that ingest the crustacean. The infection occurs when humans consume raw or undercooked crabs containing encysted metacercariae. In Mindanao, this is driven by the cultural consumption of kinilaw or raw seafood marinated in acidic juices. The researchers conducted their field sampling in Barangay Kayawan, Manay, Davao Oriental. This site was selected due to its proximity to river systems where freshwater crabs are harvested for local consumption. The study used a total of 11 randomly collected freshwater crabs. To ensure high-resolution results, the team moved beyond the tri-slide microscopy and employed a dual sequencing approach. First is the Sanger sequencing, where employed for the initial identification and high-fidelity validation of target gene sequences. And the second one is the next generation sequencing or NGS, where it is performed using the Illumina Next Sequence 1000 platform at PGC Mindanao. The NGS allowed the researchers to perform a deeper analysis of the genetic material, which provides a more comprehensive view of the parasitic load and the genetic variations within the host tissue. This transition to molecular methods is important, the way that it is very critical because traditional morphological identification of metacercariae within the crab tissue is labor-intensive and has a high failure rate in light-intensity infections. The paper then details the use of specific mitochondrial markers to achieve two distinct goals: parasite identification and host identification. For the parasite identification, the team targeted a 463 base pair or BP region of the mitochondrial 16S ribosomal rRNA and transfer RNA threonine genes. Mitochondrial DNA is utilized in this context because it possesses a high copy number per cell compared to nuclear DNA, which enhances the sensitivity of detection when identifying small amounts of parasite DNA within a large volume of host tissue. Next is for the host identification. To identify the crab species, the cytochrome C oxidase subunit 1 or COX-1 gene was targeted, specifically a 528 BP sequence. COX-1 is the standard DNA barcode for metazoans because of its high degree of sequence variation between species and low variation within species. With that, the researchers utilized the maximum likelihood or ML method for phylogenetic reconstruction. The HKY or the Hasegawa-Kishino-Yano substitution model was used for the parasite tree, and the GTR plus G plus I or the general time reversible model was used for the crab host tree. These statistical models ensure that the resulting trees accurately reflect the evolutionary distance and relationships between the Davao Oriental samples and the international reference sequences. Historically, Sundasalfusa philippina was the only recognized second intermediate host for P. westermani in the Philippines. Its molecular evidence suggests that the COX-1 sequencing revealed that the infected crabs from Manay belong to the genus Isolapotamon. Specifically, the sequences showed a 99% similarity to the Isolapotamon SP reference, referring to the accession number MT514316.1 as an example. The Isolapotamon is a genus endemic to the Mindanao region. This proves that the transmission cycle of P. westermani in the southern Philippines uses a different ecological niche than in Luzon. This expands the no transmission ecology beyond the Grapsoidea superfamily, moving into the Potamidae family. And identifying Isolapotamon sp as a carrier allows for targeted environmental surveillance in fast-flowing, high-elevation river systems in Mindanao, where this specific genus is known to thrive. The final analysis of the data provides three major factual takeaways regarding the local epidemiology of Manay, Davao Oriental. First is that the phylogenetic tree in figure 1 shows that the P. westermani sequences from Davao Oriental formed a Philippine clade. These sequences were closely grouped together and were distinct from the isolates found in Vietnam, the Vietopotamon aloyensi, and Japan, Eriochir japonica. This indicates a locally evolved and adapted lineage of the parasite. Second is that of the 11 crab samples analyzed, 5 tested positive for P. westermani DNA, representing a 45% prevalence rate. This data indicates a higher risk of zoonotic transmission to the local human population. And third, the researchers concluded that the movement of these crabs is restricted by geographical barriers like the saltwater and mountains. This then results in a focal nature of outbreaks because the crabs stay within specific river systems. The disease remains concentrated in hotspots. The genomic surveillance allows health authorities to map the specific rivers and barangays, providing a level of precision that traditional national surveys cannot provide. What we can conclude from this recent study is that only through molecular sequencing can we map these true ecological pathways and move toward the eventual elimination of paragonimiasis in Mindanao. We now expand our scope from the long flukes of Mindanao to the complex parasitic landscapes of the Visayas, specifically Northern Samar. This is where we emphasize that the molecular revolution is not limited to identifying new hosts, but is critical for quantifying the true prevalence of established pathogens. The image here identifies a schistosome, a blood fluke that represents one of the most significant socio-economic health burdens in the rural Philippines. We will specifically analyze how high sensitivity molecular tools like droplet digital PCR and RT-qPCR have revealed that our previous understanding of parasitic prevalence in these regions were statistically incomplete. The study by Vera Kuhn et al. In 2018 gives us a comprehensive molecular analysis of polyparasitism in the municipalities of Lawang and Palapag, Northern Samar. This research utilized droplet digital PCR or ddPCR to detect Schistosoma japonicum. In terms of epidemiological data, the study focused on 18 endemic barangays. Across a sample size of 412 participants, the ddPCR analysis revealed a 74.5% prevalence rate for S. japonicum. This is categorized as hyperendemic. If we examine Table 2, we see a critical breakdown by gender and age. Notably, infection intensity measured by the copy number index or CNI was highest in males, having a mean CNI of 91.7 compared to females having a mean CNI of 30.9. This is likely due to the increased occupational exposure in water-heavy agricultural work. The molecular data also revealed that S. japonicum does not exist in a vacuum. Utilizing multiplex quantitative PCR, the researchers screened for intestinal protozoa or IP and found 58.7% belong to Blastocystis spp, 21.8% Cryptosporidium spp, 19.2% belong to Giardia duodenalis, and 12.1% is Entamoeba histolytica. Statistically, 80.8% of subjects, or 248 out of 307, were positive for S. japonicum, also harbored at least one or more IP co-infections. The most frequent combination was S. japonicum and Blastocystis spp, having 62.9%. This is biologically significant because the mucosal damage and immune dysregulation caused by the schistosome eggs facilitate the persistence of these protozoans. The conclusion of this study is clear. Traditional single pathogens surveys fail to account for the synergistic morbidity caused by these concurrent infections. While the previous study addressed blood flus and protozoa, Aula et al. In 2020 utilized RT-PCR to re-evaluate hookworm prevalence in the same Samar municipalities. Historically, hookworm control in the Philippines focused on the human-specific Necator americanus and Ancylostoma duodenale. Using the primers and probes detailed in Table 1, the researchers identified an overall hookworm prevalence of 52.8% among 432 human subjects. The major breakthrough was the identification of Ancylostoma ceylanicum. Of the individuals positive for Ancylostoma spp, 26.6% were molecularly confirmed to be carrying A. ceylanicum. This species is morphologically indistinguishable from A. duodenale under standard microscopy, which is why it remained underreported. A. ceylanicum is a zoonotic hookworm, meaning it requires an animal reservoir to maintain its life cycle in the environment. To confirm the transmission route, the team sampled 33 local dogs roaming the same barangays. RT-qPCR then confirmed that 36.4% or 12 out of 33 of these dogs were positive for A. ceylanicum. This data then provides the following factual conclusions. First, the human infection is being maintained by environmental contamination from domestic animals. Second, human-centric mass drug administration or MDA programs will inevitably fail in these regions because the dogs serve as a constant source of environmental recontamination. And third, the use of species-specific qPCR targeting the ITS2 and repeat regions is the only reliable method for differentiating these zoonotic threats from human-specific ones. When we look at the two cases together, we see a pattern. In Northern Samar, we have a 74.5 schistosomiasis rate and a 52.8% hookworm rate. So the molecular evidence proves that these communities are suffering from high-intensity multi-pathogen infections that traditional microscopy simply cannot quantify. The presence of zoonotic hookworms in dogs and high IP co-infection rates in cysto patients indicates that our public health strategy must move toward one health multi-pathogen molecular surveillance model. We must treat the environment, the animals, and the humans as a single interconnected system. As we look at the future, we must ask, are we satisfied with good enough diagnostics? The future of our field lies in the integration of the tools we have discussed, LAMP for the field, ddPCR for absolute quantification, and next-generation sequencing for host parasite mapping. Looking at the image of the microscope in the background, it remains a vital tool for training and basic identification, but it is no longer the final word for surveillance. The future is molecular surveillance. This means moving from a reactive system, treating people once they show eggs in stool, to a proactive system, identifying the DNA signatures in the environment, the animals, and the hosts before massive morbidity sets in. In this slide, it summarizes the diagnostic gap that exists in the Philippines. In Palapag, Northern Samar, the CATOCATS microscopy found a prevalence of 22.9% for S. japonicum. However, qPCR found 90.2%. This refers to the study by Gordon et al. In 2015. We have been missing nearly 70% of the infected population. These invisible cases are still suffering from liver fibrosis and are still shedding eggs back into the environment, continuing the cycle of infection. Next is from the study of Machong in 2017, specifically focusing on the school children in Laguna, referring to the table 2, the discrepancy is just a start. microscopy found a 33.8% prevalence for soil-transmitted helminths or STHs, while the qPCR found 78.3%. So why does microscopy fail? First is that the parasites don't shed eggs every day. If the patient provides a sample on a non-shedding day, then they get a false negative. Second, as noted in the Aula paper, the hookworm eggs lice or break apart very rapidly after defecation. So if the KK slide isn't prepared and read within 30 to 60 minutes, the eggs disappear. And molecular tools detect the DNA even after the egg has sliced. Third is that the traditional Kato-Katz method uses only a tiny smear, approximately around 41.7 milligrams. So if the infection is slight, the chances of an egg being in that tiny smear are statistically low. So summing up what has been discussed so far, only through molecular sequencing can we map the true ecological pathways. We are currently underestimating our national parasite burden by a factor of 3 to 4. We cannot reach our 2030 elimination targets using 22.9% accuracy when the reality is 90.2%. So to conclude, we have looked at the Ricard case study in Mindanao, which discovered a new host, and the summer studies which proved that our current prevalence data is fundamentally flawed. The molecular revolution is about bringing equity to our rural health units. By adopting tools like LAM and genomic surveillance, we provide every Filipino, whether in a remote barangay in Manay or a rural school in Samar, with the highest standard of care. We are no longer guessing, we are sequencing. So here are the following literature cited in this reporting presentation, and thank you for your attention.